So in this video, we're going to talk about respiration and all the different reactions that we get that result in the production of ATP. So in the diagram here, we can see a mitochondrion in the cytoplasm of a eukaryotic cell. The line on the outside is the cell membrane, so we just can use the labels. And we can see glycolysis is happening at the top in the cytoplasm. So we can do a bit of annotation on this one. We know that the glucose enters the cytoplasm through carrier proteins by facilitated diffusion. We know that the glucose is phosphorylated twice using up 2 molecules of ATP, and that will make hexose bisphosphate. Now we know hexose bisphosphate is quite reactive, it's quite unstable, so it's going to split into 2 molecules of what we call triose phosphate. So triose means 3, so it's a 3-carbon organic molecule with phosphate attached. Now the triose phosphate, and we would have to put triose phosphate in the real exam, not TP, becomes oxidized to pyruvate. So the oxidation is the removal of hydrogen, and the hydrogen gets picked up by a coenzyme called NAD to make NADH. During this oxidation step at the top, there's also some release of energy, which allows the transfer of a phosphate group directly onto ADP to make ATP. So for every triose phosphate, we know 2 molecules of ATP get made by substrate level phosphorylation. Now, if we follow the diagram, should be from point B to point C, we do know that pyruvate can get across both mitochondrial membranes, the outer and the inner membrane of the mitochondrion. And when it's in the matrix, we know that we get the link reaction. So if we look at point C, this is the product at the end of the link reaction called acetyl coenzyme A. But we know point D must be one of the other products that gets released. Now D cannot be FADH, even though it does have an arrow coming from the Krebs cycle, because actually the only coenzyme to get reduced in the link reaction is NAD to become NADH. So D must be NADH because that's made in both the link reaction and the Krebs cycle reactions. Now we did say acetyl coenzyme A is the acetate is the 2-carbon compound that's attached to coenzyme A. We call it acetyl coenzyme A. Acetyl coenzyme A carries in the acetate group into the Krebs cycle, and we get a series of oxidation reactions, and we also get some ATP being made again by substrate level phosphorylation. Now if we have a quick look at point E, E must be carbon dioxide because we can see it gets across both mitochondrial membranes. From the cytoplasm, it can diffuse simply across the cell membrane, and therefore it can be released from the cell. And we know carbon dioxide is small, we know it's not charged, it's not polar, and we know it's lipid soluble. So if it's being made in the matrix from the link reaction and the Krebs cycle, it can simply diffuse out of the mitochondrion, out of the cell as a waste gas. Now if we have a look at F on the left, we should be able to see that it starts from outside the cell, it diffuses across the cell membrane into the cytoplasm, and then across both mitochondrial membranes. And the arrow for label F is actually pointing at the electron transport chain, and we know oxygen is the final electron acceptor that's needed to pull electrons through the ETC, combine them with some protons to make molecular water. So F would be oxygen, which is the final electron acceptor. So once we've annotated this, we can then move on to the actual questions themselves. Okay, so A, B, C, D, E, and F, we can now just quickly pop in. So A is the 3-carbon triose phosphate, spelt out, not abbreviated. B was pyruvate, which is the end product, the 3-carbon molecule from the end of glycolysis, which is actually made in the cytoplasm. C was acetyl coenzyme A. Please do not try to abbreviate that. They will not accept acetyl CoA. They actually want the full name of that one. D was reduced NAD or NADH is okay, or you could put NADH/H+. They will not accept reduced NADH though, so it's just NADH or reduced NAD. E was the small lipid soluble carbon dioxide, which can simply diffuse out of the cell, and F diffuses the opposite way into the mitochondrion, into the matrix, and that's oxygen. Now the next question says, give a reason why ATP is used up in the process of glycolysis. So it's really important to get the keywords correct. So we know ATP supplies phosphate ions, and the phosphate ions are used to phosphorylate glucose to make hexose bisphosphate. Now hexose bisphosphate we know is very reactive, unstable, and that's the molecule that's going to split into two molecules of triose phosphate. Okay, so we know the process of glycolysis happens in the cytoplasm of all cells using glucose as the main substrate for these reactions. And it's really important to learn a model answer for glycolysis. So this is AO1, but then we could be asked to apply a bit of this knowledge to an AO2 question. So glucose is phosphorylated using up two molecules of ATP. The phosphates, one of the phosphate ions from each ATP is transferred onto glucose, and that's going to make a molecule called hexose bisphosphate. Now this is quite reactive, it's unstable. It's going to split into two molecules of triose phosphate, which are both obviously 3-carbon organic molecules. Each of the two triose phosphate molecules is then oxidized, which we know means it loses hydrogen. The enzyme would be dehydrogenase, and it's going to transfer the hydrogen from the triose phosphate onto a molecule of NAD to make a molecule of NADH. So NAD is basically a taxi for hydrogen. We call it a coenzyme because it needs to pick up the hydrogen that was released from the oxidized triose phosphate. Now, during this oxidation of triose phosphate, we also know that some energy is being released, and it's going to allow the direct transfer of phosphate onto ADP to make two molecules of ATP per triose phosphate. Now, we call that substrate level phosphorylation because a substrate molecule, in this case it's triose phosphate, is being broken down, it's being oxidized, and it's releasing some energy, which allows us to build that bond between the phosphate and the ADP to make the ATP directly. There's no electrochemical gradient needed for this production of ATP. Now the net products of glycolysis are two molecules of ATP net are made per glucose, two NADH are made, and two molecules of pyruvate are made in the cytoplasm. So part B to this question gives us a little bit of information about an investigation that was carried out into the production of pyruvate. And what they've done is to take some cells and they've physically broken up the cells by a process called homogenation, and they've released the cell components, including the nucleus, the mitochondria, and the cytoplasm, and they've separated these components based on a process called centrifugation. So if you take the cell components and spin them at relatively low speeds, the nucleus pellets at the bottom of the centrifuge tube, and you can collect the pellet and isolate the nuclei. The liquid that's left over is called the supernatant, and then you can spin that at slightly faster speeds, and then you will pellet out the next dense organelles like the mitochondria, for example. And if you collect that pellet and isolate the mitochondria, what you're left over with in the supernatant is, for example, the cytoplasm. So we can see very clearly that the pyruvates are only made in the cytoplasm, and we want to explain these results. So the answers are that glycolysis only happens in the cytoplasm because the enzymes for glycolysis are located in the cytoplasm, which are needed, for example, to make the triose phosphate and then to oxidize the triose phosphate into the 3-carbon pyruvate. So we can only get pyruvate where we have the enzymes necessary for glycolysis, which is only in the cytoplasm. So this follow-up investigation looked at the effect of different types of inhibitors on the reactions within glycolysis that happens, as we know, in the cytoplasm. And it does say in the table that if you add inhibitor A, the final product would be triose phosphate, or inhibitor B, the final product would be glucose, or inhibitor C, the final product would be hexose phosphate. So this indicates that the inhibitor must be inhibiting the reaction after the production of these three molecules. So for example, at the top with inhibitor A, that must inhibit the reaction after triose phosphate has been formed, because triose phosphate cannot now go on to be oxidized. So this inhibitor A maybe inhibits triose phosphate dehydrogenase, because the final product that accumulates is the triose phosphate. Likewise for glucose, if we get an accumulation of glucose, it means the next reaction in glycolysis cannot be happening, it's being inhibited. So maybe inhibitor B is going to inhibit the enzyme that would normally phosphorylate the glucose to make hexose bisphosphate. So if we cannot get phosphorylation, we're going to accumulate the glucose in the cytoplasm fraction. Now for inhibitor C, we can see hexose bisphosphate has been formed, which indicates that phosphorylation must have taken place of the glucose, but that the hexose phosphate itself cannot split into the two molecules of triose phosphate. So inhibitor C is inhibiting the breakdown, the degradation, the splitting of hexose bisphosphate. So we can have one mark for each of the three inhibitors. So mark point A is inhibitor A inhibits the oxidation of triose phosphate into pyruvate. So triose phosphate is the final product that accumulates. Inhibitor B inhibits the phosphorylation of glucose, so the enzymes that normally add the phosphate onto glucose. And inhibitor C inhibits the splitting of the hexose bisphosphate into triose phosphate, which indicates there's an accumulation of the hexose phosphate. So the next part of the question talks about a large increase in the glucose concentration would reduce the action of inhibitor B. Now we did say inhibitor B does block the phosphorylation of glucose because glucose starts to accumulate. But as we increase the glucose concentration, we know glucose is a substrate in order to be phosphorylated. It's a little bit like increasing the substrate concentration above the inhibitor concentration, which indicates that that reaction is more likely to proceed. It's more likely that the glucose will actually bind with the active site of the enzyme, which will allow phosphorylation of the glucose compared to the inhibitor. So this is one way of testing if an inhibitor is a competitive inhibitor or a non-competitive inhibitor. Whereas if we actually increase the concentration of substrate, which is glucose, we're going to dilute out the effect of a competitive inhibitor. So for the second point, a large increase in the substrate concentration, which we know is glucose, allowed the rate of reaction to be higher, reach its maximum level. There's more enzyme substrate complexes, and that's the evidence we need to say it's a competitive inhibitor. So in this question, we're given a nice diagram. It's labeled, so we're going to use the labels where we can. And it actually tells us that it's an inner mitochondrial membrane, and there's two big proteins that we can see involved in oxidative phosphorylation. And the smaller protein in pink is actually an electron carrier that carries electrons down the ETC. So the white protein on the left, it's called cytochrome BC1. That is actually proton pump number 2. We know it spans the inner membrane of the mitochondrion, and we know it does actually pump protons into the intermembrane space, and it uses the energy that's lost from the electrons as they move down the electron transport chain. Now on the right hand side, the sort of yellow orange looking protein, that's proton pump 3, and it's called cytochrome C oxidase. And the pink cytochrome C in the middle is the electron carrier that carries the electrons between proton pump 2 and proton pump 3. So proton pump 2 is called cytochrome BC1. The electron carrier between the two proton pumps is called cytochrome C. And then proton pump 3, of course, it wants the electrons from cytochrome C. So it has some enzyme activity, which is why it's called cytochrome C oxidase, because it oxidizes the cytochrome C that was originally reduced as it carries the electrons through the inner mitochondrial membrane. So we're just going to use the labels as much as we can. Now, part A says cytochrome oxidase or cytochrome C oxidase is the final protein in the electron transport chain. It is an enzyme which transfers the electrons to a molecule of oxygen. And we know oxygen is the final electron acceptor, So the oxygen would actually pull the electrons through cytochrome C oxidase at the bottom, proton pump 3, and it would combine them with some protons, and that's going to make molecular water, which is the final product, isn't it, of oxidative phosphorylation. Now it says state the final product of this reaction in the mitochondrion. So we can just state H2O or water. Now the next part of the question says the cytochrome C oxidase molecule is different in different species. The diagram below shows the enzyme from a cow, which is this one on the left, versus a bacterium. The yellow or the orange shaded section, though, in each represents the polypeptide chains, which are very similar in both organisms. That means this primary structure, this polypeptide chain, is highly conserved, which means this part of the protein has a very similar tertiary structure because it's a very, very important protein or enzyme, if you like, in aerobic respiration. And it does tell us this is the area of the enzyme which binds to the cytochrome C electron carrier in order to pull the electrons from that carrier and into and through the two enzymes that we can now see on the picture. Suggests why the shaded regions are similar in all organisms, highly conserved. Well, it is actually the active site of the cytochrome C oxidase enzyme, which we know is proton pump 3, and therefore the active site has to be a complementary shape to the shape of the cytochrome C electron carrier in order to pull the electrons away from cytochrome C, which therefore oxidizes cytochrome C. So it's not saying the substrate, it's actually using the name of the electron carrier, cytochrome C, for the second point. Now, we could also have had an alternative answer, which says the genetic code or the DNA base sequence in the DNA that codes for the primary structure of the polypeptide chain is going to be similar in both organisms, which means the DNA base sequence is not mutated. It's very, very highly conserved, which means the sequence of amino acids in the polypeptide chain that makes these two parts to the enzyme, cytochrome C oxidase, is very conserved. It's very similar. The primary structure of the polypeptide chain is very similar, which gives a very similar tertiary structure and therefore a very similar activity. So as the cytochrome C electron carrier binds the active site because it's got a complementary shape to cytochrome C oxidase, it then says that the cytochrome C will actually detach once the electron has been removed from the cytochrome C. And it says, please explain, well, why is this the case? So essentially, cytochrome C is the substrate, and when it's formed an enzyme substrate complex, it loses its electrons, so it's oxidized, which actually changes the shape very slightly of cytochrome C. So the shape of this molecule, electron carrier, is not now complementary in shape to the shape of the active site of cytochrome C oxidase, and therefore it detaches from the active site, leaving the active site free to be used once again. So obviously the removal of electrons from cytochrome C is oxidizing cytochrome C. It is changing its shape, so it can no longer form the enzyme substrate complex with the cytochrome C oxidase enzyme. So the third part to this question, part C, it does tell us that cytochrome BC1 is a proton pump that transfers protons or pumps protons, H+ ions, into the intermembrane space, which is where they can start to accumulate. Then it says a chemical called antimycin can actually bind to cytochrome BC1, or proton pump number 2, and block its action. So it's going to stop it from transferring electrons down the ETC. If there's a backlog of electrons, there's no loss of energy to activate the proton pump to pump in protons into the intermembrane space. So it's asking us to say explain what would happen to the pH of the intermembrane space when the antimycin is present. So because fewer protons are being pumped into the intermembrane space, because the electrons can't move through the ETC anymore, there's no loss of energy, they're not being activated, the pH would actually start to increase. So you could say it might become more alkaline over time if it goes above pH 7, or it certainly would become less acidic with time. So the next part of this question is another AO2 application question, and it says oligomycin A is another type of chemical which this time acts by inhibiting protons from passing through ATP synthase. So we all know that protons normally go from a higher concentration through the stalk particle and therefore through ATP synthase to a lower concentration. So they go out of the intermembrane space through the ATP synthase and back into the mitochondrial matrix. The question says, explain why the presence of this chemical in a human causes high levels of reduced NAD to form in the cell. So when the protons come from higher to lower through ATP synthase, stalk particle, they normally combine with electrons and oxygen to make molecular water. So if there's no protons coming through ATP synthase, it means that the oxygen is not pulling the electrons through the ETC, because the oxygen has to be used up with the electrons and the protons to make water. And there's a backlog of electrons along the electron carriers in the electron transport chain. This is a bit of a problem because any new NADH which is made from glycolysis or the link reaction or the Krebs cycle, that needs to be oxidized at the start of the electron transport chain. So NADH would normally be oxidized to NAD plus hydrogen, which splits into protons and electrons. Now the electrons would normally move through the ETC through redox reactions via electron carriers, and then the protons can get pumped into the intermembrane space. So if we have a look at the answers then, that number one, the protons do not bind to electrons and oxygen at proton pump 3. Please do remember this helps normally to keep the protons fairly low in concentration in the mitochondrial matrix as they get used up to make the water. The electron carrier system would then stop, which means any new NADH cannot be oxidized at proton pump number 1. So NADH cannot pass its electrons and its protons to the ETC. And the second part of the question says, why would lactate begin to accumulate in the blood if high levels of reduced NAD started to form? Well, lactate is made inside animal cells, such as muscle cells or liver cells, when the oxygen is not available, and it's equivalent to anaerobic respiration. So pyruvate, we know, is reduced to lactate, and that's going to use up the NADH because the hydrogen from NADH is transferred to pyruvate to reduce it to lactate as part of anaerobic respiration. And this is important because the NAD that's just been regenerated allows glycolysis to continue in the cytoplasm of the cell, and we know that allows a small amount of ATP to continue to be made in the absence of oxidative phosphorylation, which can't occur if the ETC is blocked, and that would be by substrate level phosphorylation. So in this question, it's going to talk about substrate level phosphorylation, and it says it's the simplest, oldest and least evolved way to make ATP. So quick annotation on this one then, some reference to substrate level phosphorylation. We know it happens in glycolysis, and we also know it happens in the Krebs cycle in the mitochondrial matrix. So this is where ADP and a donor phosphate group normally combine to make ATP directly, and there's no electrochemical gradient needed for this. This is very different from oxidative phosphorylation. There's no electron transport chain, no electron carriers, no proton pumps involved in the production of ATP in this way. So it says at the end, the energy released during the conversion of one molecule into a different molecule is used to combine ADP and PI in order to make the high energy bond of ATP. So the first part says, describe the position of the high energy bond referred to in the paragraph. Well, the high energy bond, which is formed through condensation when ADP and PI are combined, is between the last phosphate group and the second to last phosphate group. Bear in mind, we know adenosine triphosphate has 3 phosphate groups. So the bond between the terminal or the last 2 phosphate groups of ATP. The second part says, suggest why substrate level phosphorylation is referred to as the simplest and oldest way to make ATP. Well, this is where we can bring in the annotation. So we know this does not involve the electron transport chain. It does not involve the electrons moving down a series of electron carriers to activate proton pumps. So there's no electrochemical gradient. That means it does not involve the stock particle, which we know is found on the inner mitochondrial membrane. Protons can move through this by facilitated diffusion. That doesn't happen in this case. No electrochemical gradients, so there's no pumping of H plus ions into an intermembrane space. And then because there's no electron transport chain, it means oxygen is not needed to pull the electrons through the ETC as the final electron acceptor. So no oxygen is actually required for substrate level phosphorylation. We just need a substrate molecule being broken down. For example, triose phosphate, it gets oxidized, it loses hydrogen, and some of the energy that's being released in that reaction can combine a phosphate, which is actually transferred from triose phosphate directly onto ADP to form the high energy bond in ATP. So the third part to this question asks us to draw 2 arrows, and we are going to label them A and B on the diagram to show the 2 steps where substrate level phosphorylation does take place. So if we look at the picture, we can see hopefully that the 3-carbon sugar in glycolysis is going to be oxidized. We know that 3-carbon sugar with a phosphate on it is triose phosphate, and we know it loses hydrogen by dehydrogenase. We also know the phosphate from triose phosphate is combined with ADP to make ATP. The second part would be the 5-carbon, we call it alpha ketoglutarate. As that is dehydrogenated or oxidized, it's also decarboxylated, and it's going to make another 4-carbon compound. It's actually called succinate. And during that step, there's energy being released to directly form ATP in the mitochondrial matrix. So part 4 to this question asks us to give the number of ATP made by substrate level phosphorylation at each of the conversions shown in the previous diagram, but it does say per glucose molecule, and this is aerobic respiration. So point A we know is glycolysis. We know as triose phosphate is oxidized to pyruvate. There's actually 2 molecules of ATP that get synthesized per triose phosphate. Now, because glucose is a 6-carbon molecule, you get 2 triose phosphates, and each triose phosphate gives 2 ATP. So the gross number of ATP is 4 from a single molecule of glucose in glycolysis. We do know that 2 of the ATP are invested in the investment phase to phosphorylate the glucose, but the gross number is 4. And then the alpha ketoglutarate, the 5-carbon compound in the Krebs cycle, that gives 1 ATP per cycle. But because we get 2 Krebs cycles per glucose, because don't forget we had 2 pyruvates and therefore 2 acetyl coenzyme A's, we're going to get 2 molecules of ATP directly. Now this question is fairly straightforward. It's just AO1 recall. So we know the link reaction happens in the mitochondrial matrix, which is the fluid organic matrix in the middle of the mitochondrion. We know it involves pyruvate, and we know there's 2 things that happen. Pyruvate is oxidized or dehydrogenated by dehydrogenase. That's why we get 1 NADH, and we know it's also decarboxylated by decarboxylase. So the 3-carbon pyruvate would form the 2-carbon acetate. Okay, so the 2 types of enzymes then are dehydrogenase, that's the oxidation reaction, which reduces NAD to NADH, and the decarboxylase removes the carbon atom as a molecule of carbon dioxide. So part C to this question is talking about the experiment we did in the lab using methylene blue. And it says that the methylene blue, when you add it to cells or to mitochondria, acts as an artificial hydrogen acceptor. So it mimics the role of NAD. So when dehydrogenase oxidizes a substrate and removes hydrogen, normally that hydrogen is transferred onto the NAD coenzyme. But methylene blue, mimicking NAD, can also become reduced by the activity of dehydrogenase. So when it becomes reduced, it decolorizes and the blue color disappears. So we can get an idea of the rate of respiration by timing how long decolorization occurs. So in this experiment, they've isolated mitochondria from liver cells. And once they've broken open the liver cells and they've centrifuged the mixture, the mitochondria have then been isolated from a pellet. But once they've isolated the mitochondria, they've put them in ice-cold isotonic buffers. So the question at the bottom says, once they've been isolated, why are they initially suspended in the ice-cold isotonic buffer solution? Well, when you homogenize and break up cells or tissue, very often lysosomes which are found inside the cells are also broken open. So there might be some hydrolytic enzymes within the mixture, things like proteases, nucleases, etc. So we put the mitochondria on ice in this suspended liquid to reduce the activity of any potential hydrolytic enzymes to stop further digestion of the mitochondria themselves. Isotonic means the water potential of the matrix of the mitochondria will be the same or about the same as the water potential of the suspension liquid. So there's no water potential gradient between the liquid and the inside of the mitochondria. So there's no net osmosis. Therefore, the mitochondria can't take in water, so they don't swell and burst, or they don't lose water by osmosis, so they don't shrivel up due to that loss of water. And the buffer, we know that maintains a pH, so it's a pH buffer. So any enzymes inside the mitochondria, such as the enzymes of the link reaction or the Krebs cycle or oxidative phosphorylation, they will all occur at the optimum pH that we've set the buffer at. So in terms of making a model answer then, it's really important that we've got some experimental details written down. The ice-cold liquid or suspension liquid slows the activity of potential hydrolytic enzymes, for example, from ruptured lysosomes. Isotonic maintains the same water potential inside and outside the mitochondrion, so there's no net osmosis in or out, so there's no osmotic lysis, for example, of the mitochondria, or they don't shrivel. And the pH buffer maintains the optimum pH for all the enzymes like dehydrogenase and decarboxylase inside the mitochondria. Now, if you start with isolated mitochondria, please remember these are the organelles. We haven't got any cells in this experiment. So we've actually broke open the cells by homogenation and isolated the mitochondria. It says, why is pyruvate the substrate added to the mitochondria, but not glucose? So you'd only add glucose to a sample if it was the cytoplasm, because that's the site of glycolysis. So all the enzymes of glycolysis are found in the cytoplasm. And there's no carrier proteins for glucose to get inside the mitochondria anyway. And even if glucose could get inside the mitochondria, because there's no enzymes for glycolysis, nothing's going to happen to the glucose. Now we know, obviously it says at the top, glucose enters glycolysis in the cytoplasm, but not the mitochondria. And if we add pyruvate, we know that can actually enter the mitochondria through both the outer and the inner mitochondrial membranes. And once pyruvate actually gets into the matrix of the mitochondrion, it then enters the link reaction, where it gets dehydrogenated and decarboxylated. So number one, glycolysis is in the cytoplasm. That's where all the enzymes for that glycolysis are. Glucose is the substrate for that. Pyruvate has to be added to mitochondria because it's got carrier proteins that get inside whereas glucose can't. And then it's got the link reaction in the matrix. Okay, so the last part of the question gives us some data to start to evaluate, and it's asking us to draw one conclusion from the experimental data in the table. So we're given 3 trials at each of the different temperatures tested, and they've calculated the mean value. So it's the mean seconds for methylene blue to decolorize, and then we're given some standard deviations above and below each mean value. So if you look at the trend of the mean values, hopefully you can see that 102 seconds is the lowest of the time points. That indicates that the methylene blue is being fully decolorized the fastest at 40 degrees C. So it suggests that 40 degrees C is the optimum for the dehydrogenase enzymes inside the mitochondria, for example, from the link reaction and the Krebs cycle. Now, the other data that we really want to look at is the standard deviations. So we can see at 10 degrees C, there's a mean value of 332 seconds. So it's obviously taking longer for methylene blue to decolorize, but there's a lot of variation in the 3 trials that they've measured. So because the standard deviation is plus 48.6 above 332, or it's minus 48.6 beneath 332, that indicates that the mean value is less reliable because there's so much spread of data away from that actual mean value. Now, we can also look at the other standard deviations, and they are a lot smaller. So, for example, for 40 degrees C, the standard deviation is plus 3.5 above 102 or minus 3.5 below 102. So because that is a small standard deviation, there's less spread of data away from the mean value. There's less variation in the 3 repeats, and so the mean becomes more reliable. The other point to notice is that 165 seconds at 30 degrees C, the standard deviation is plus 6.5 or minus 6.5, and there's no overlap between those standard deviation bars and the standard deviation bars for 40 degrees C. So 102 plus 3.5 or 102 minus 3.5, the SD bars do not overlap with the SD bars from 30 degrees C. Likewise, the 102 mean value the standard deviations do not overlap with the standard deviations from 50 degrees C, which is the 155 second mean underneath. So the first mark point then is 40 degrees C is the optimum for the dehydrogenase enzymes inside the mitochondria. 10 degrees C is the least reliable of the temperatures tested because of the great standard deviation. It's much higher than the other standard deviations, and any correct use of the data with respect to the reliability of 20 to 60 degrees C. So all these SDs are much smaller. And I would also incorporate the idea that because there's no overlap in the standard deviation bars from 30 to 40 to 50 degrees C, these mean values are significantly different from each other. And the 102 mean value is the significantly lowest value of all of the means.